Hello, Dr. Swandi Shamano here. Welcome to Punches Biology, or Punch Biology in the Face. Today, we'll look at the human digestive system. The human digestive system consists of the alimentary canal and the accessory organs. Accessory organs include the liver, the gallbladder, pancreas, and salivary glands. The alimentary canal is a 9-meter long tube that extends from the mouth to the anus. It's therefore important to differentiate the digestive system from the alimentary canal. The alimentary canal is part of the digestive system. The alimentary canal is a tube running from the mouth to the anus. The liver, the gallbladder, pancreas, and salivary glands are not part of the alimentary canal, but they are part of the digestive system. They are accessory organs of the digestive system. Digestion is the process where large insoluble food molecules are broken down into smaller soluble molecules that can be absorbed by the body cells. Using the diagram, take some time to study and master the organs of the digestive system and any other structures involved. We'll now look at the stages of food processing. There are 5 different stages of food processing. The first one is ingestion. Ingestion is the taking in of substances such as food and drinks into the body through the mouth. Digestion, like we've said, is the breaking down of large insoluble food molecules into smaller soluble food molecules. So digestion can be mechanical or physical, which involves the breakdown of food substances into smaller pieces without chemical change to the food molecules. Then secondly, there is chemical digestion, which is the breaking down of large insoluble molecules into smaller soluble molecules. The next stage is absorption, which is the movement of small food molecules and ions through the walls of the intestine into the blood stream. Here the food has been digested, the insoluble has been converted to the soluble, and therefore can be absorbed and therefore can be moved from the intestines into the blood stream. Next is assimilation. Assimilation is the incorporation and utilization of absorbed food molecules into the cells of the body for energy, growth, and repair. So the body, after it has absorbed the food, it will now utilize the food. It will utilize the food to generate energy, utilize the food for growth, utilize the food for repair. Finally, there is egestion. Egestion is the removal of undigested or unabsorbed food substances as feces through the anus. Please note that egestion is not the same as excretion. Excretion is the removal of waste products of metabolism. Egestion, on the other hand, is the removal of undigested or unabsorbed food substances that cannot be digested, food substances that cannot be absorbed. For example, if you consume or take in a plastic material, that plastic material will still come out through the anus as feces or part of feces. The plastic cannot be digested. So that is egestion. Let us now look at the different organs which are involved in the process of digestion. We'll start with the mouth. Digestion begins in the mouth, and the space in the mouth is called the buccal cavity. In the mouth, we have teeth. The teeth break down the food into smaller pieces, and this is called physical digestion, or it's an example of physical digestion, where we said in physical digestion or mechanical digestion, we're breaking the large food substances into smaller food substances. This makes it easy to swallow and also increases the surface area of the food for the digestive enzymes to work better. In chemical digestion in the mouth, the salivary glands secrete saliva, which moistens the food and makes it easier to swallow. The saliva also contains salivary amylase, the enzyme called ptyalin, an enzyme which breaks down starch into maltose. The optimum pH for salivary amylase is 7. The tongue in the mouth rolls the food into a bolus, which is then easily swallowed. We are now in the esophagus. The esophagus is a muscular tube that leads to the stomach. It is made up of layers of smooth muscles, the two layers, the external layer called the longitudinal muscle and the inner layer called the circular muscle. These muscles help propel the food down the esophagus through a process called peristalsis. So peristalsis is the rhythmic, wave-like muscular contractions in the wall of the alimentary canal which pushes the food down the esophagus. This is also true for the intestines. So peristalsis occurs in the esophagus and in the intestines, where food is propelled forward. We are in the stomach. This is where proteins are digested. In the mouth, we digested starch. In the stomach, we're now digesting proteins. The walls of the stomach contain gastric glands. So these glands secrete a juice called gastric juice used in food digestion. The gastric juice contains hydrochloric acid and pepsinogen. The pepsinogen is the inactive form of pepsin. Gastric glands also secrete mucus, which protects the stomach walls from digestion by the pepsin. The HCl in the gastric juice 1. Changes the inactive pepsinogen into the active form pepsin. 2. It provides an optimum pH for pepsin to work better. The pH is about 2. The hydrochloric acid also helps kill germs and bacteria. The hydrochloric acid also stops the activity of the salivary amylase by denaturing it. Moving down to the small intestine. The small intestine is the final place for the digestion of food. Here we digest the starch, we digest the lipids, and we digest the peptides. The small intestine is divided into 3 parts. The duodenum, which is the first part, the second part called the jejunum, and the third part called the ileum. Food is moved through the small intestine by peristalsis. The pancreas produces pancreatic juice, an alkaline solution containing trypsinogen, pancreatic amylase, and pancreatic lipase. So remember we said the pancreas is one of the accessory organs of the digestive system. So the role for the pancreas here is that it produces a juice called pancreatic juice, and this juice contains trypsinogen, which is an enzyme that will be used to digest the peptides, pancreatic amylase, which is an enzyme that will be used to digest starch, and pancreatic lipase, which is an enzyme that will digest the lipids. Pancreatic juice enters the duodenum through the pancreatic duct. All enzymes in the small intestine have an optimum pH under alkaline conditions. But remember, the food is just coming from the stomach where it was acidic. How then does the pH change to alkaline? Bile, an alkaline greenish yellow fluid, is produced by the liver. The liver is one of the accessory organs of digestion. The liver is one of the accessory organs of the digestive system. So after the bile is produced by the liver, it is stored in the gallbladder. When there's food in the duodenum, the gallbladder contracts, pushing the bile into the duodenum through the bile duct. The bile serves a number of functions. The bile contains sodium bicarbonate, which helps neutralize the acidic chyme as it comes from the stomach. The food coming from the stomach is called chyme, and it's acidic, remember, because the gastric glands produce the hydrochloric acid, making the pH acidic. But we've said the enzymes in the duodenum work well under alkaline conditions, so we need to neutralize the acidity, and this is neutralized by the sodium bicarbonate, which is found in the bile. The bile also contains bile salts. The bile salts are important in a process called emulsification. Emulsification is the breaking down of large fat droplets into smaller fat droplets. This helps increase the surface area for the enzyme lipase. Absorption of food. Food is completely digested in the small intestine. The jejunum and the ileum function mainly to absorb nutrients and water. The small intestine is adapted for this role by having an inner wall with large circular folds, finger-like projections called the villi, and each epithelial cell on the villi has smaller projections called microvilli. The villi are only one cell thick so that food molecules can easily diffuse into the bloodstream. These adaptations increase the surface area of the small intestine, resulting in a large surface for absorption. Within each villus is a network of capillaries and a small vessel called a lacteal. Nutrients are absorbed across the wall of the small intestine and the capillaries or lacteal. The lacteal transports fats away from the small intestine, while the capillaries transport sugars and amino acids. The large intestine, also called the colon, has the function of absorbing the remaining water, amino salts that have not been absorbed by the small intestine. The undigested food matter moves along the large intestine by peristalsis and mainly comprises of cellulose, which is indigestible to humans. The waste matter ends up at the rectum where it is stored before it can be eliminated from the body through the anus. The elimination of undigested food material is called egestion. This wraps up today's lesson, but before you go, take a look at some review questions.